Baik, Dr. Arifin, time is yours. You can deliver in English or campur-campur lah di sini. Jadi selesai nanti silakan yang mana yang boleh disampaikan dan Yasmin akan membantu untuk menayangkan share screen-nya. So, Bismillah, kita akan mulai. Silakan, Dr. Arifin.
Baik. Bismillahirrahmanirrahim. Alhamdulillahirrahmanirrahim. Wassalatu wassalamu ala asyrafil anbiya wal mursalin wa ala alihi wa sahbihi ajma'in. Bismillah sadri wa yassir li amri wa uqdatan min lisani yafqahu qawli. Subhanaka la ilma lana illa ma 'allamtana innaka antal alimul hakim wa la hawla wa la quwwata illa billahil aliyyil azim. So, first of all, I would like to thank Professor Dr. Sri Hadi Ayoni, my beloved friend and teacher actually, for inviting me to deliver this topic to the students. Really happy to have this session and share about this topic. I would like to also thank the Dean, Bapak Dr. Amrozi, for sending the official invitation for this lecture. So welcome to the students and also I think also some staff from the APB. Welcome to this lecture. So I'm not really very expert, just some experience doing some research on microRNA, which is one of my research interests since 2012. The topic that I will deliver today is on the role of microRNA in the epigenetics mechanism of a few diseases. Right. I will deliver, inshallah, maybe campur-campur as Pak Yoni mentioned earlier. But I will like to jump. But I'm afraid that some of Malay and Indonesian bahasa Melayu and bahasa Melayu Malaysia and Indonesia maybe some different in meaning. I encounter some experience on that. So that's why I mean, as much as possible, I'll try to deliver in English to avoid the different meanings lah. Sometimes I have experienced some misunderstanding just because of difference in meaning of words when I was in Indonesia. Yeah. So try to deliver in English, yeah, and maybe maybe I will use also some Malay language. All right. So the role of microRNA in the epigenetic mechanism of diseases. My name is Muhammad Arifin bin Kaderi. I'm currently an associate professor at the Department of Biomedical Science in Kuliah of Allied Health Sciences, International Islamic University, Malaysia, in Kuantan campus in state of Pahang. So a little bit on my academic background. I received my PhD in molecular and clinical genetics from Uppsala University in Sweden. That was back 14 years ago in 2010. And before that, I received my MSc, Master of Science in Chemical Pathology in University Science Malaysia in 2004. And before that, Bachelor of Biomedical Science in University Kebangsaan Malaysia in the year 2000. In 2012, 2014, I did my postdoctoral research in the World Health Organization, International Agency for Research on Cancer. I was working on the genetic susceptibility of in nasopharyngeal carcinoma. And in 2015, I received the Mary Curie Fellowship under the European Union Commission. And one highlight in my career also is the research that I performed at the Pusat Studi Satwa Primata, IPB, that was under the sponsorship of USAID, United States Agency for International Development, and also CO Hun. I think Bapak Prof. Yoni is also one of the key person in One Health in Indonesia, And there's a lot of collaboration also between universities and countries under the One Health consortium. So we hope also to build a lot of collaboration in issues regarding human and animal health. And this is one of the pictures that I mean, that's precious to me. Me and my teacher and my research colleagues at PSSP in 2018. Right? Maybe you still, I mean, some of these lecturers and also researchers at PSSP, Pak Imong and also Pak Us and Ibu Silmi. I think they are still there. Okay, next. So back to the topic of today. These are the some of because usually when we deliver lectures, we are supposed to tell the students what are their learning objective of the lecture in general. Right. So in short, these are the learning objectives. Number one, to define microRNAs. What are the microRNAs and their biological roles? And then explain the concept of epigenetics and its relevance to microRNA. And then we will explore how microRNAs act as biomarkers in human diseases and discuss example of microRNA epigenetic biomarkers in cancer, cardiovascular disease, and neurodegeneration. And we will also highlight challenges and opportunities in clinical application. If we have time, I will also try to share some potential research and also findings regarding microRNA in veterinary medicine, inshallah. All right, next. Okay. All right. So before we go directly to microRNA, just maybe I think maybe the postgrad students has learned a little bit on epigenetics. I'll just summarize and share a very short introduction on epigenetics. Right. So the definition of epigenetic is the heritable changes in gene function. So the keyword there is gene function, and without altering the DNA sequences. So it's different. Epigenetic is different from mutation, right? Okay. So because the changes in epigenetics does not directly involve changes in the DNA sequence, right? The genome is still intact, the same, but the function of the gene is different, is modified because of the epigenetic mechanism. So there are three key epigenetic mechanisms that we know today have been researched, has been studied. So number one is DNA methylation, and then histone modification. Example there is acetylation and methylation. These are mechanism that changes the structure of the DNA. Right, it doesn't change the sequence of the DNA, changes the how the DNA is being read. Right, especially at the region of promoter and also the gene itself. I will not go deeper in DNA methylation and histone modification because our topic today is more on microRNA. And microRNA belongs to the group of what we call as non-coding RNA. Right. So there are actually several other non-coding RNA, such as long non-coding RNA. There are several others, but one of the most widely studied is the microRNA. Right. Okay. Next. Okay. So this is the if we learn about in molecular biology, I think we learn about the central dogma of molecular biology, which is the process that involve DNA to RNA to protein. So originally we are taught, I think, during our degree and also during courses involving molecular biology or genetics, the process is a one-way process. We have our genome, and within our genome there are DNA, and there are segments in our DNA that are genes that encode for the instruction for every single activities that we have, that we need to carry on or to carry out during our lifetime, right? And this instruction is located at our DNA, in our gene especially. And this instruction will be copied and through the process of transcription. And the copy of this instruction is called RNA, mainly the mRNA, right? Okay, and then this mRNA will be read and will be translated. Okay, will be read and will be translated into protein, and this protein is the functioning unit for all of the activities that we need to do during our lifetime. All right, and involving when we blink our eye, we move our fingers. These are all the activities that occur due to the interaction between different protein complexes, right? And to produce that protein complexes, we need the instruction in our DNA. And we were taught, when especially when I was younger, much younger. I think the students now are very young and yeah, very young, vibrant, and things like that. So during those time in the 90s, we were taught that this process is one-way traffic from DNA, RNA, protein. But then later on, we also learn about a process called reverse transcription that occurs mainly in the prokaryotes and also in viruses. Very rarely occur in mammalia. Um, but also, we learn that this process is not that straightforward. There are, as we learn more and more about biology, there is also different mechanism on how this process is being controlled, right? And um, especially after the completion of the Human Genome Project, after we learn about more about our genome, knowing that only 1% of our genome actually encode for encode for protein. So what about the other parts of our genome? Right? So we learn more and more that other parts of our genome are actually encoding for different system, different function that control how the gene is being used or being applied towards the function. So one of the discovery was on the discovery of microRNA. Okay, and we learned about the process called genetic regulation, which, as I mentioned earlier, about chromatin remodeling. We also learn about mRNA splicing. We also now learn about RNA degradation. And one of the process is through microRNA, right? So the process of transcription and translation is being, there are lots of mechanism where this is controlled, and one of the way of controlling this is through microRNA. And why do we need that? Because, as human being, we interact with the environment, and not always that, it's not all the time our genes were transcribed and translated into protein in a very stagnant ways. So we need to adjust, okay, how the gene functions due to the changes in the environment, right? For example, like the function of our skin. For example, when we go to when we live for several years in the cold temperature areas, our skin tend to become a little bit more lighter, because the expression of melanin will be reduced, right? And one of the way this is done is through the function of microRNA in reducing the level of melanoma in that is being produced. All right. Okay. So that is in short, and I give the source there. The where I think student, you can read a little bit more on the function of microRNA in the epigenetics. All right. Okay. Next. All right. So we will look into now the microRNAs. What are the microRNAs and how it functions? All right. So these are the next slide, please. Okay, these are the overview of microRNA that we will look through. So first, definition and discovery, and then the structure and biogenesis, and then mechanism of action through the RNA-induced silencing complex, and also the key role of microRNA in the post-transcriptional gene regulation. So in short, the definition of microRNA. So microRNA are small, they are very small, endogenous and non-coding RNA. So the size of microRNA is around, on average, around 22 nucleotides. So, we learn in molecular biology, the size of human genome is around 3.2 billion, right? Base pairs, right? So of our DNA, imagine that is that big, our genome, right? But microRNA, the size is very, very small, only 22 nucleotide, very small. And this small nucleotide can function to regulate gene expression at the post-transcriptional level. So after the mRNA has been produced through transcription, the microRNA can bind to this mRNA. Right? So, I mean, please don't mix up between mRNA and microRNA. Sounds a little bit same, and the symbol is also quite similar, but they're different. mRNA is the one that will be translated into protein, while the microRNA will bind to the mRNA, right? And then it causes modification or changes in the function of the mRNA. All right. So so this microRNA will bind to the especially the UTR, the 3' UTR region of the mRNA, and it can cause the mRNA degradation, or it can cause us the translational repression, reducing the translation of the mRNA. All right. So I will go through the other point later on. Next one, when we look into the figure regarding the function of the microRNA. Sorry. So next slide, please. So yeah, microRNA is a big thing this year, especially, right? So recently, very recently, I think it was back in um, September or October. Sorry, September or August. It was declared that this year's Nobel Prize is awarded to these two scientists, right? Victor Ambrose and also Gary Rafkin, right, for the discovery of microRNA. And interestingly, most of their research actually was performed in the animal, the worm, right? The C. elegans worm. Okay, they discovered the function of one of the earliest discovered microRNA called Lin4 and Lin7, and discovered how they function in regulating the post-translational gene regulation, right? And they studied this also later on in higher animals, including human being, right? So yeah, microRNA is a big thing this year, right? Okay. So next, please. Okay. So with regard to microRNA biogenesis, this is the summary of the function of microRNA. So so yeah, microRNA is actually being produced in the nucleus from the gene that encode for for this microRNA itself. And once it leaves the nucleus, it is in the form of this hairpin, as you see in number one there. All right. So we leave a nucleus in this form, and then there will be it leave it's transported by exportin 5, transport it out of the nucleus, and then an enzyme called dicer. There's an enzyme that will cut the microRNA, especially it will cut at the loop there. If you think about the hairpin, right? There's a loop. So this dicer will cut the microRNA close to the loop, so that we have the double-stranded microRNA, and then later on, a process will occur that will separate the double strand and it will later on become a single strand short RNA. These are what we call as the mature microRNA. And this microRNA, in plant cell especially, this microRNA will bind perfectly to the mRNA, and this will lead to degradation of the mRNA. So imagine the cell will produce mRNA, okay, for its function, but depending on the environment, this mRNA may not may not require to be translated into protein. So the process that will inhibit or avoid this translation process is through microRNA function. That's in plant, usually the binding of the microRNA to the mRNA will lead to the degradation of the mRNA itself. But in animals or in animal cells, especially including in human, usually the microRNA bind not in a very perfect way, that will not lead to the degradation, but will lead to the reduction in the translation of the mRNA. So the amount of protein, the level of protein that will be produced from that mRNA will be reduced. Okay, that is the regulation of the translation that will occur due to the function of the microRNA. Right. Next slide, please. Okay, this is the more detailed story about the biogenesis of microRNA. So as I said, microRNA, there are parts in our genome that encode for the gene of the microRNA, and then DNA polymerase II will read and will produce the pre-microRNA in the nucleus, in this form, the hairpin form, and then through the process of the enzyme ROSHA and DGCR8, it will process this pre-microRNA into pre-PRE microRNA, and then it will go to the cytoplasm. In cytoplasm, the dicer will bind to the pre-microRNA, and it will produce the what we call as guide strand. This guide strand will bind with other protein, and it will produce a complex called RISC. So RISC will bind to another protein called argonaute, and this complex, microRNA RISC is the one that will produce the mature RNA, mature microRNA. This mature microRNA is the one that will bind to the mRNA, right? So in human especially, and in mammals, the binding of this complex, microRNA argonaute complex, will induce the translational inhibition of the mRNA, so that the outcome will be reduction in the production of the protein that is being produced, right? Next, right? Okay, so we move on to the section 2, roles of microRNA in physiology in normal situation. So microRNAs are critical regulators of gene expression and play diverse roles in maintaining physiological homeostasis. They control various cellular processes by fine-tuning protein production at the post-transcriptional level, ensuring appropriate responses to the environment and developmental cues. All right. So this is epigenetics. Our body will always adapt to the environment, and the way we adapt, one way of adapting is through the function of the microRNA. Right, next. All right. So yeah, these are some of the key function of microRNA. Right. Number one in cell lineage specification. Right. When the embryo is being produced, right? Right. So microRNA plays very important role in regulating, the development during embryology. All right. So microRNA regulate the expression of genes that determine the fate of the cell. All right. Example: miR1 and miR133 control muscle cells differentiation by targeting transcription factors involved in myogenesis. All right. The process of producing the muscle cells during development in the embryo is controlled by these two microRNAs. All right. Involvement of these two microRNAs. Of course, there are other mechanism of controlling the process, but two of the important mechanism is through this miR1 and miR133. All right. So, and then microRNA also involved in the stem cell maintenance and differentiation. Right? For those interested in doing embryology, this is also an important function of the microRNA. MIR 290 through to MIR 295 cluster are essential for maintaining pluripotency of embryonic stem cells. Okay, so you know the embryonic stem cells are the cells that will develop into various other cells, that produce bones and muscles and connective tissues. And the determination of the development of this embryonic stem cell is controlled by this group of microRNAs, right? And also, LED7 is microRNA that promote differentiation by suppressing stemness-related genes. So LED7 is very important in the development of embryo, that it will induce the maturity of the cells, right? So it will reduce the level of mitosis of the cells, and then determine that the cell is going towards being a mature cell, right? Okay, and also microRNA is important in the neurodevelopment, the development of nerve cells, okay, saraf, yeah, right? miR124 and miR9 can regulate neuronal differentiation and maintain brain development by targeting neuronal cells, neuronal genes in neural cells. All right. So yeah, miR124 and miR9 is very important in the function of development of saraf, okay, the nerve cells, right? So there's a research actually doing a study on these two MIR to see whether modulating the expression of these two genes can also modulate or regulate the development of nerve cells. Right. Next. All right. So in the adult cells, microRNA is also still important, and one of the way microRNA play roles is through cell cycle regulation and apoptosis. Right. microRNA maintains the balance between cell proliferation and cell death. Right. So in biology, we always we have to keep in mind that for a biological system to be normal, there must be check and balance, right? And one of the way of doing this is through the function of microRNA. So miR17 through miR92 cluster promote cell proliferation by targeting tumor suppressor gene like P10. All right. So we know that to develop cells, okay, to make more cells, we need to reduce the function of tumor suppressors. I think for people who are interested in doing research in cancer, we always have to keep in mind about the balance between the oncogene and the tumor suppressor gene, right? Right. So if the cell want to grow, it needs to reduce the function of tumor suppressor gene, right? So that the oncogene will be activated, and that will lead, in general, that will lead to development. But if that is not controlled, it may lead to development of cancer, right? So there's a need to control the oncogene also. So that is, always they need a balance between the two group of genes, the oncogene and also the tumor suppressor gene, right? So in this case, MIR 17 through 92, they are pro development, pro proliferation. They target the tumor suppressors, example like P10, okay, they bind to P10 mRNA and we reduce the development of this tumor suppressor protein. And that will allow the oncogene to be activated, right? But on the other hand, MIR15 and MIR16, they induce apoptosis, right? So induce apoptosis by downregulating anti-apoptotic factors like BCL2. All right. So yeah, I forgot to mention also, not only oncogene and tumor suppressor gene, there's also another factor that influence the development, which is the apoptosis, right? And the induction of cell death, right? So MIR15 and MIR16 induce cell death or apoptosis by binding to the BCL2 mRNA, and then it downregulate the anti-apoptosis factor of the BCL2, and that will allow the cell to go through normal cell death, right? And that will reduce the risk of developing tumor and cancer. All right, next. Okay, some microRNA are also involved in the immune regulation. So microRNA are crucial in both innate and adaptive immune responses, right? For those, when we learn immunology, okay, we learn about the two arms of immune responses, which is the innate and also adaptive immunity, all right? Okay. In innate immunity, okay, the MIR146A modulates the TLR to like receptor signaling and inflammation by targeting these two genes, TRAF6 and IRAK1, right? So these genes are important in the innate immunity cells, like for example, like basophil, neutrophils, and things like that. All right. So yeah, they modulate, and these cells, yeah, basophils and neutrophils and macrophage, especially, right? They're involved in the general immune responses, right? And they are the ones responsible for the induction of inflammation, producing histamine and things like that. So MIR146 control these cells, all right? And then adaptive immunity. In adaptive immunity, when we learn immunology, adaptive immunity function especially in the production of antibody, as well as the function of cell-mediated immunity through the T cells. All right. So MIR155 promote the differentiation and function of T helper cells, Th1 and Th17, and also B cell maturation. So B cell is the one producing the antibodies, yeah? And the function of this cell can be controlled by the MIR155, all right? And also MIR21. So MIR21 is one of the most studied MIR most studied microRNA in human. So miR-21 modulates immune tolerance and inflammation, right? The level of miR-21 is important, the balance is important to induce immune tolerance, okay, to determine that the cell will not attack other cells, lah, right? So we don't want our B cell to produce antibody that will attack our own cells, and we don't want too much of this cell to induce inflammation, because that will be detrimental to the health of the individuals. And miR-21 is important in controlling this function. All right, next. In metabolism, microRNAs regulate metabolic pathways, including lipid metabolism, glucose homeostasis, and energy balance. Example is miR-33. They control the cholesterol and lipid metabolism by targeting genes like APCA1 and CPT1A. While miR-122 is a liver-specific microRNA that regulates fatty acid and cholesterol synthesis. Okay, these are key microRNA that, if you like to do studies on them, especially on a topic of metabolism control, modulation. So perhaps this miR would be interesting to you. So I have also one study. I collaborate with one study doing work on this miR, and to see how the supplement or natural product can control the production of this miR, especially in the fat cells, right? The research is still ongoing towards the end, but inshallah in 1 or 2 years we can publish the work lah. Okay. Because certain diet, certain nutrient, especially from our from our plants, can actually modulate the function of this microRNA that is involved in the production or the function of the fat cell, adipose cells, right? Next. Okay, and in cardiovascular system, microRNA are critical for heart development in the embryo, and then in adults, microRNA is important modulator of vascular integrity and function. So example is miR-1 and miR-133 regulate cardiac muscle development and contractility. And in vascular function, miR-126 promote angiogenesis by targeting negative regulator of VEGF signaling. So the higher the miR-126 will promote the angiogenesis, will promote the production of the formation of the blood vessels. Okay, and this is important, especially if you are doing research on cancer, because we know cancer will activate the development of blood vessel that will supply nutrient to it. So by reducing the function, the expression of miR-126, we hope that we can avoid, we can prevent the production of VEGF. Okay, so and VEGF, we know that vascular endothelial growth factor is important in the development of the blood vessel. We want to stop the development of blood vessel, so we can do that through inhibiting the miR-126. All right, and then miR-125, miR-145 control the vascular smooth muscle cells differentiation. Right? Okay, next. And in the nerve cell, in the neural function, so microRNA contribute to synaptic plasticity, learning, memory, and neuronal survival. So example is miR-132 promote synaptic plasticity. We know that this synaptic plasticity is important in Nerve, they need to build connection, okay, between one nerve to the other to develop, okay, the neuronal system. So yeah, MIR132 promote this plasticity and the development, induction of the development of connection between nerves, right? Apa saraf berhubung kan? Bila saraf berhubung, kita jadi lebih pintar, insyaAllah, right? And one of the way that is it's controlled and is promoted is through the MIR132, right? So MIR134 regulates the dendritic spine morphology. So for those who are learning about neuroscience, so this dendritic spine is important also in the delivery of signals through the nerves, as well as in budding, in building a new network also, right? A new, for the nerve for the neuron to branch out. Okay, it is done through the dendritic spine. All right. So the morphology of this dendritic spine is regulated by MIR134. So if you are interested in doing research on nerve cells, I think these two microRNA would be very interesting, right? And then maybe you can study about how certain nutrient can increase the level of these two microRNAs, right? And then it will hopefully induce the um, function of the neuron, as well as in the reduction of dementia. Okay, as the country, I think, develop and people live longer, I think the issues of the dementia is also increased, and we want to ensure that the synaptic plasticity and also the health of the dendritic spine is maintained, and maybe we can do that through promoting the function of these two microRNAs. All right. So next slide, please. Okay, so microRNA is also important in wound healing. So microRNA influence processes like inflammation, angiogenesis, and tissue remodeling. MIR21 again promotes fibroblast migration and proliferation during wound healing, right? And MIR29 modulate extracellular matrix remodeling by targeting collagen production, right? So this two MIR is important in the wound healing, but also we have to keep in mind this process, if not being controlled, may also lead to cancer again, all right? So yeah, we need to control, we need to have to get the balance, okay? We don't want to promote the MIR21 and 29 too much, and will maybe lead to metastasis and full-blown cancer. But at the same time, if we can induce them, induce their function, it may be good for the wound healing, all right, and production of collagen. Okay, next. And in aging, okay, microRNA play dual roles in promoting and suppressing aging-associated process. MIR34A and MIR29B are associated with cellular senescence and aging phenotype, while LED7 family microRNAs promote healthy aging by regulating insulin signaling and inflammation. Okay. This is not particularly my field, but I think you're interested in the study of aging and senescence. This microRNA may be interesting to you, right? Okay. Next slide, please. Okay, we move on to section 3, microRNA as biomarkers in human disease. This is one of the field that I'm doing my research currently with postgraduate students, right? The biomarker research. Okay, so before we talk about the microRNA itself, we learn about biomarker, the indicator, one molecular component that can give us information about situation that we are studying, right? Certain diseases or certain condition, okay, that can be indicated by the level of certain protein or certain component, cellular components. All right. So when for those who are doing biomarker research, we know that the important attributes of a good biomarker would be the sensitivity. Okay, a biomarker should be sensitive so that small changes in the level of the biomarker is indicative of the condition. Okay, whether it is positive or it is negative. All right, and then the biomarker must be specific. The biomarker, when it is increased in specific cell, it is indicative of whether it is going towards pathological condition or whether it's clear or not. This is also important, specific, and then it must be stable. Okay, and one of the new important attributes of biomarker is non-invasiveness. The more invasive the biomarker, for example, in order for us to study those biomarker, we have to take sample um, from tissues. For example, in cancer, usually before this, to study or to get clinical information about whether someone is having the tumor is through biopsy. So biopsy is painful usually, right? Um, to take a tissue from an yeah, from the body to do biopsy, usually it's painful for the patient. So that will make the patient avoid doing that test. So if we can have a biomarker that can be taken without doing this painful test, that would be much better. All right. So non-invasiveness is one important factor to be a good biomarker. All right. And for that, microRNA can be an ideal candidate. Number one, the factor that is important for that is microRNA is detectable in biofluids. So you can take blood. It's minimally invasive from the vein. Okay, you can take blood from the vein. You can take saliva. This is what we did lah mostly in our research. We take saliva from the patient, right? And then we can also take urine. Okay, not very non-invasive, and we can measure the level of certain microRNA from this biofluid, right? So they are either non-invasive or minimally invasive in the case of blood, in the case of venous blood. And then microRNA is very stable in biofluid. So before people have attention on microRNA, usually we try to study the gene expression through studying mRNA. Then we know that that's very difficult because mRNA is unstable, especially if it's in biofluid, right? mRNA, even though even after we extract the mRNA, we cannot store them for long because it starts to degrade. So very unstable. But microRNA is very, very stable. One of our study, as published quite recently, shows that microRNA is stable in this biofluid, even though the level of other RNA is very, very fluctuating, right? So, yeah, stable in biofluid. And then microRNA is disease specific, okay, in terms of its profile, its expression profile, depending on the condition of the disease. So it is disease specific expression profile, indicative of the situation. And then it's rather easy to detect and to quantify. Right? There are several technologies that we can choose from to study depending on the needs of our research. right? We can, if we want to study only 2 or 3 microRNAs, okay, we can use simple real-time PCR, quantitative PCR to study that. And if you want to study, if you want to screen for lots of microRNA, if we don't have any ideas how the microRNA, what microRNA is important for specific disease that we want to study, and we want to screen for as many microRNA as possible, we do have technology to do that, either through next generation sequencing, or as we did in our lab, we did through the what we call as PCR array. We screen around 800 microRNA and see which one would be the most potential candidate that can be the biomarker for the condition. And then one important key point for microRNA is we can detect microRNA at early stage, right? Especially in cancer. Okay, during the development of the cancer, before before it developed into cancer, it's still at the neoplasia or in tumor stage. Okay, the tissue starts to release microRNA, and this microRNA will go to the blood, to the peripheral blood, and we can we can detect this microRNA in the venous blood. All right. And yeah, microRNA, as I said, it's non-invasive or minimally invasive. And the upregulation or downregulation of the microRNA is associated with key pathways of diseases, and it has potential as therapeutic targets. When certain abnormal microRNA increase in the blood or in the tissues, that is indicative of pathological condition, and we can use the microRNA as the therapeutic targets. Okay, so we can develop certain therapeutic strategy to reduce the development of the disease. All right, next. Okay, so yeah, and so microRNA has tissue specific expression pattern. Many microRNA exhibit tissue specific expression and making them highly valuable for identifying the source of disease. Example is miR-122 is liver specific and strongly associated with liver diseases like hepatitis, fibrosis, and hepatocellular carcinoma. miR-133 and miR-1 are muscle specific and is associated with cardiovascular disease and muscular disorders. And there are resources in the internet that we can access for free, as these are the link that I provided, and we can look, if we, for example, we want to study diseases related to, for example, muscle tissue or liver. So we can look at the list of the microRNA that is commonly expressed in this type of cells, right? So we can access it directly through this this database. Okay, that is open source, right? So yeah, next. Okay, can we move on? Okay, stability in biological fluid. Yeah, microRNA are remarkably stable, as I mentioned earlier, in variety of biofluids, including blood, saliva, urine, cerebrospinal fluid or CSF. Of course, to get the CSF, it is quite invasive, but a certain condition we may need to get samples from CSF. And there's also microRNA is stable in breast milk. We have one group in our university working on studying the microRNA in breast milk. All right. And yeah, microRNA can resist degradation. So they resist degradation by RNases, and because as we know, mRNA is very sensitive to RNAs, and RNAs is everywhere. So that's why mRNA is very unstable. But microRNA is stable because it is protective. It is protected in the form of complex we call as RISC. And microRNA in the circulation, in the blood, is encapsulated, it's covered by extracellular vesicle called exosome. Okay. And it can also be protected by what we call as apoptotic bodies and also high density lipoprotein binding of the mRNA through the HDLs. All right. And this stability ensures reliable detection and quantification, even in minimally invasive or non-invasive sample. We can take from the blood and it's stable in the blood, and we can measure them, and it's quite stable for a certain time. All right, next. Okay, can we move on? Okay, and then easy detection, as I mentioned earlier, we can use quantitative RT-PCR. And this is a gold standard for microRNA quantification, especially if we already know what microRNA we want to study. Right? We can do also microarrays and next generation sequencing. This is high throughput profiling of microRNA expression. And you can just take samples from many patients. For example, in our case, 42 samples of nasopharyngeal carcinoma, and then we send to the lab and they do the next gen sequencing for us. All right, and they give us the profile of the microRNA expression. And a new technology now is through through the technology of droplet digital PCR or ddPCR that enables absolute quantification of microRNA in complex samples. I've not done this personally, but there are also group in our university that is doing droplet digital PCR for microRNA quantification. And yeah, it's the cost for running this technology is, I think, reducing now, right now. And NGS used to be very expensive, but nowadays we have a new technology called nanopore, which is a little bit, it's more affordable for us to do. Right now, also there are services that we can send our samples in China. I think there are several labs that provided services for next generation sequencing with quite reasonable price. Right. Next. Okay. And as I mentioned, microRNA expression has this specific expression. Example, MIR21 again is overexpressed in many cancer. MIR146 and 155 is regulated in inflammatory disease and specific for that disease. All right. Next, I think due to the limitation of the time, I will go quite fast. Yeah, some of the slide I will skip. Next, please. Okay, so microRNA is apa ni is good for early detection of diseases. So microRNA, as mentioned earlier, microRNA are often dysregulated at early stage of diseases and sometimes before the clinical symptoms. Right? This is important, especially in cancer research, because we want to search for that biomarker that can be detected even before the disease actually has manifest the symptom, because we know that in cancer, if symptom is already manifest, that is an indicator that the cancer is already at the later stage 3, stage 4, and usually at this stage is very very difficult to manage lah. Very difficult to treat the patient if they are already at stage 3 and stage 4. That's why we want to detect at early stage. So example here is MIR29 is an early biomarker for liver fibrosis, and detecting high level of microRNA 29 will enable early intervention before irreversible damage occurs. MIR126, a level in circulation can indicate endothelial dysfunction and earlier marker for cardiovascular diseases. Before the patient develop heart problem, we can detect the problem at early stage, so that we can intervene at early stage before the disease has already in full blown condition. All right. Next one. Okay, so yeah, microRNA has certain microRNA has direct association with key pathway in diseases. So microRNA regulate critical cellular pathways involved in diseases mechanism. So example is like cancer, right? So miR34 targets P53 signaling. So you know, P53 is a key indicator for late stage of cancer, right? So, but detecting MIR at MIR34 at early stage, it will be indicative of how P53 level would increase or decrease over time. All right. In cardiovascular disease, MIR133 affects cardiac hypertrophy pathways. All right. So, the higher MIR133 mean the cardiac hypertrophy pathways will be activated. So we can use this as a target for treatment. Right. And then in neurodegenerative disorder, MIR29 and MIR124 are implicative of Alzheimer disease. So before the Alzheimer disease is developed, we can measure the level of MIR29 and MIR124. If we see the pattern of increase, then this is indicative that the patient is moving towards Alzheimer, so that we can start the intervention at early stage before the Alzheimer disease has fully developed. All right. So yeah. So this association allows microRNA to serve as biomarker indicator for molecular dysfunction. So that knowing this at early stage before the disease has already developed fully will allow us time to decide how we can intervene to stop the development of the disease. Right. Next. Right. Okay, so that the general information that I can share regarding the microRNA and its related to diseases. I just want to share some of the key studies, key publication that we have developed, that we have produced over time. So this one is the first paper that we use as the guide for our research lah. The Magdalena Wozniak is my colleague at the WHO IRC. She and her group actually produced this paper on circulating microRNA, meaning that microRNA that we can get from the venous blood and also from urine, and they become the biomarker for early detection of non-small cell lung cancer. All right. So this publication is one of the key guide that we use in the study of microRNA. Okay. Next. This is one of the first paper that we published in a Thai journal, differential expression of MIR101 and MIR744 in nasopharyngeal carcinoma. So we have been studying nasopharyngeal carcinoma in Malaysia and trying to find a biomarker for early detection of this NPC. Because most of the NPC in Malaysia is diagnosed at early stage. So we want to screen for the high risk group to detect whether they will develop NPC or not. And if we find any possibility or risk of developing, we can allow that can allow the clinician to intervene. So here we studied this two microRNA. We found that one of the microRNA is upregulated and one is downregulated in NPC, and this can serve, when the imbalance occur, it is indicative of the development of NPC at early stage, all right, before the disease already developed into stage 3 and stage 4. So yeah, so we can detect this at stage 1 and stage 2, and it's much easier to manage the patient when we detect at that stage lah, right? So usually nasopharyngeal carcinoma, if we find that the patient is having cells, the benign tumor, so the surgeon can go and when they cut off the area of the NPC, take out the tumor, the benign tumor when it's still very small, and that will avoid development of the cancer, right? So yeah, that's our collaboration with the clinician on that. Okay, next. Okay. All right. So in this study, this was published in Indonesian journal. I think it's UI journal, Makara Journal of Health Research. I think it's maintained by University Indonesia. Yeah. So in this we report, how the microRNA is actually stable, even though the quality of the RNA is quite poor. All right. So meaning that we can still use microRNA as biomarker, uh, even though the quality of the sample is quite not too optimum. All right. So we cannot do that with mRNA, but we can still do this with microRNA. MicroRNA is still stable. All right. Even though the purity and the yield of the RNA is low lah. So that shows that microRNA is very, very useful, right? Even though the level, the the quality of this sample is not good lah. No, not to say not good, but not optimum. Okay, all right. And next one is the thesis of my PhD student. Next slide, please. Yeah, this is the thesis of my PhD student on circulating and salivary microRNA in NPC. This student has already graduated, and we're still trying to publish a couple of papers more from this study. But I think I can share key result from his study lah, Dr. Azmir, in the next slide. Okay, so we can see here, this is his abstract. And if you read here, in the end, we actually screen around 800 microRNA. And then we do a lot of analysis using bioinformatics. And for the statistical analysis, in the end, we found two microRNA that is most potential, that has the highest potential of serving as the biomarker for detection for early diagnosis of NPC, okay, which is HSA MIR 150 and MIR 205. So we zoom in to these two microRNA, and okay, this can be confirmed later in later study whether they still can serve as biomarker in a larger population of nasopharyngeal carcinoma patients, right? So 4 years of doing this work, and alhamdulillah we come up with something, but still in research, okay, one result after hard work still not enough. We need to still do further studies lah, right? To confirm whether these two microRNA can really serve as early detection biomarker for this disease. So if it's confirmed, then I think it will contribute a lot in terms of screening and surveillance of the risk group population in Malaysia. And maybe we can extend this to Indonesia later on. We can, if we have a collection of sample from patient, we can also measure these two microRNA and see whether we can confirm that these two microRNA can serve as important and effective biomarker for the early detection of the cancer. Right. So next slide. I think I'll stop there first. If we still have time, I can share some of the potential research or potential finding on microRNA in the veterinary medicine research. But I think stop there first. and maybe open for the floor for questions. Prof, over to you.
Yes, thank you very much, Dr. Arvin. Alhamdulillah, yeah, it's very nice presentation, and I think this sharing is very, especially for the students. I think very important and very valuable information for you all. That most of them, Dr. Arifin, they are trying to, of course, to doing the research by themselves, and your experience and your sharing will be good. What to say, arahan lah ya, macam contoh, like examples for them. That the research is quite in the medicine is quite very terbuka, open, and quite so many things that we can do. Baik, silakan para student yang ingin bertanya, you can ask bahasa Inggris boleh, bahasa Indonesia pun boleh ya, and you
Insyaallah,
can
bisa.
yeah, you can write or you can just ask directly, please. Yes, Atwisto, you have any question? Silakan. One question from Nur
Okay.
Anissa. Boleh terbaca, Dr. Arifin?
Boleh,
Atau
saya boleh baca. Okay, I've been
quite long?
talking.
Yes,
Okay.
okay.
Hmm.
Very, very nice question, but quite long.
Yeah.
Yeah, okay. I think the question
Okay.
is in the end of the.
Yeah.
Ya, boleh. Silakan, Dr.
Yeah.
Arifin.
That's true. One microRNA may have different function lah, and different effect on several target genes, and yeah, it needs a lots of bioinformatics pre-analysis before we embark on full blown, or we go to wet laboratory, wet experiment on certain microRNA. Um, yeah, it's a long process. I think we can discuss further if you're interested. I can bring the people, the expert. I'm not an expert in bioinformatics myself, right? But usually, what we start from is we work together with bioinformatician. We go through these databases to find candidate lah. That is if we want to target one microRNA. Right? But if we want to do screening, we can screen. For example, there is now services. Kaizen provides panels for certain diseases. If you are interested in specific diseases, for example, like inflammation or cancer, I work more on cancer. So we can discuss with the technical support from Kaizen. They will advise us on what microRNA that is the most potential for specific diseases, and we can screen for those microRNA. So maybe we know that in total there will be around 1,000 microRNA, human microRNA that has been identified and known what functions they are. But for certain diseases, there may be around 100 or less, right? So we don't have to screen all of them. So in terms of cost, that will be more cost effective. So yeah, usually before we embark on full blown experiment, we do pre-analysis first. All right. So we see the panels. We see whether specific microRNA is really specific for that condition that we want to study, or it will be that special there are panels of interesting microRNA, so that we don't have to study all of them. Okay, so I hope that answered the question. All right. So yeah, pre-analysis is important. So yeah. So using it nowadays, I think current generation is very lucky because there's a lot of these supports, the companies providing the support that we can advise us how to do about before we actually do the research itself. So if you're interested, we can further discuss, and I can bring in the more technical people lah. Okay. Because I myself I'm not the one that doing the full-time research in the lab. We have the people who are more knowledgeable on the technical part. All right. Okay. So the second question, I hope
Second
that
question.
answers your question, yeah.
It is okay, Nur Anissa. Boleh ya? Yes, now we go to the second question by Yasmin. I think the
Okay.
question is why mRNA 21 ya
Yeah.
boleh memiliki function yang beragam.
Hmm.
Apakah dia sangat powerful or yes.
Yeah. Okay. miR-21. Yeah. One of the reason is because at the level of at the level of mRNA itself, the protein that is involved here is also important in several mechanisms. Right? So this miR-21 binds to this mRNA, and because this mRNA functions functions in the production of a protein that has also multiple function. Right? So that is why this miR-21 has different effect in different condition. Right? So miR-21 is the one most studied, but usually after miR-21, we have to continue with the more specific microRNA. miR-21 is interesting as a start to see whether um, uh, it can serve as biomarker for this condition, but then we have to confirm with other microRNA to confirm the condition. All right. So miR-21 is a good start, but usually we have to continue with other more specific microRNA. Right. Okay. Hope that
Okay,
answers the
Yasmin.
question.
I think yes ya. Okay.
Thank you,
Next we
Yasmin.
have Dr. Danang. Ya, Dr. Danang, please.
Baik, terima kasih Prof. Yoni atas kesempatannya. Thank you, Dr. Arifin, for your nice presentation. As previously mentioned in your presentation, that microRNA is one of the non-coding RNA in which they cannot translate it into proteins, right?
Yeah.
So in this case, I would like to ask your opinion, or based on your experience, if we compare to messenger RNA, which one is better, or which one is best represent the, for example, disease condition? I mean, between the microRNA and messenger RNA, which one is better to represent the disease condition? For example, in metabolic disease, based on your experience. Thank you.
Okay. Thank you very much for the question. Very, very interesting and important question. In terms of, especially in metabolic disease, of course, when we talk with the molecular biologists, okay, there's different school of thoughts. Yeah, some of them they say protein is the most important. Some says mutation is more important. So yeah, different ideas and different opinions. We know that the end product that is functional is always protein, but mRNA is indicative, and gene expression, especially messenger RNA, right, is also indicative. But we know that there are limitations about messenger RNA. Sometimes we see the upregulation and downregulation of mRNA may not be truly representative. Many times when we do research, we see that this mRNA is upregulated, but then when we move on to the protein, we see that there are discrepancy in the results between the protein expression and the mRNA expression. That's why we go now to the deeper level, which is mRNA, something, we want to provide explanation of what is the discrepancy between the mRNA and the protein. It may be due to the function of microRNA, right? So we can start either way. We can start either by doing protein study, can do Western blot or other more high-tech technology to study the protein, and then we go back to try to explain what happens at the molecular level, right? Or we can do the other way around. Okay, we study the microRNA, right? So if we have specific candidates of microRNA that we want to study based on literature, it will be much easier. Maybe one or two microRNA or a little bit less than 10 microRNA that we want to study. Do qPCR on that microRNA, see whether this microRNA level is higher. So and then we follow up with the messenger RNA study, whether the result will be concurrent or tally with the result. And then I think if we want to get the full story, the three things, if we want to study mechanism, I think doing microRNA and then mRNA and then protein will provide the full story. But for example, if we want to design a study for master's program, right? So student would not have a lot of time to do that, and then technology limitation. So I think to me, microRNA is the easiest to perform because it's stable and it doesn't require postgraduate research. We are limited in terms of the student are not expert yet in terms of doing lab works. So gene expression through messenger RNA may be may produce very unconclusive result because handling of the sample mRNA require handling mRNA messenger RNA requires very careful and very confident in terms of technical management. And doing protein research, again, it's very tedious, take a lot of time, and again, doing protein work, protein can be degraded quite easily. But I think doing microRNA, because microRNA is stable, between the three, and from my experience, between the microRNA, mRNA, and protein, I think microRNA is easiest to do because it's stable, right? And especially when our lab is not like in European lab where everything is properly designed and properly managed and things like that. microRNA I think easier for master's level and PhD level. But I think if you want to do a long-term research, we want to confirm the mechanism, it's best if you can do all three parts, which is microRNA, messenger RNA, and also protein. But if you want to look for biomarkers, I think for me, my experience for biomarkers, microRNA is a very good potential because it can be taken from non-invasive sample, convenient for the patient, and it's stable. I think these two factors are the main factor why microRNA is a very good candidate for biomarkers. Okay, all right. Hopefully that answers.
Yeah, that's what I mean, Dr. Arifin, because messenger RNA expression is not always represent the protein expression. So how about microRNA? Is that enough if we just, for example, detect the microRNA and that's it, we get the real condition, for example?
Yeah. If we study mechanism, I think not enough. microRNA alone, I think it's not enough, but I think it's a good start to indicate the story, because mechanism usually it's we want to get the full picture of the story of the condition. So if you want to start with microRNA, I think that will be a good start. So that after microRNA, we can follow up with the gene expression and the mRNA, and then we want to do the protein level, so that we have the full picture of the mechanism. And I think if to get the full picture, I think it requires at least 2 or 3 PhDs lah, right? And yeah, quite a lot of time, but yeah, that's a good thing. We can have more project for the postgraduate students, right? So yeah, so I think for mechanism, I think the 3 parts would be helping each other lah, to get the full picture. Right? Okay. Ada question lagi?
Dr. Nang?
Mhm. The research
Freeze,
related
I
to
think.
microRNA.
Yes,
Yeah.
the question from Nur Anissa again.
Mhm. Permission provide information global funding. Yeah, this is
Yeah.
it's a tricky question. Yeah, it's a good question. We have the same problem in Malaysia also. Um, for us, I think in Malaysia, mainly the resource of funding is a national grant. We have what is called as fundamental research grant. Yeah, well, it's still
From the
it's quite
Ministry of Education, like that? Dr.
Ministry
Refik?
of Higher Education.
So
But
I
it's very
think the similar
competitive.
thing.
Yeah.
Yeah.
But the good thing is the good thing about this microRNA research is that because of the Nobel Prize thing, so microRNA becomes like a hot topic currently, right? Doing microRNA research, I think it attract attention of the funder. I'm not sure about the Indonesia. What is the provider, the main provider for research? But in Malaysia, yeah, there are several new grants have been approved on microRNA research. One of the reason maybe because the Nobel Prize thing helps a lot on that. Okay, global one. I think for global grant. Sometimes we have to tackle. I mean, I think Prof. Yoni also have experience with USAID. I think we have to tackle what are their interests. The American usually their interest is on infectious disease, because they are concerned about disease from our region transmitted to their people in the US. So there are several source of funding on infectious disease. For me myself has received some grants from USAID on infectious disease. For fundamental research on mechanism microRNA, it's quite challenging for us because usually the high impact research is more impactful in their lab, right? Especially for in cancer, for example, for popular cancer like breast cancer, like for example melanoma, things like that. There are many big labs in the US and in Europe doing that. So for us to compete with them is quite difficult. But maybe certain cancer or certain condition that is specific for us, they may not be interested in funding it unless we find collaborators in the US. I think another way, another trick is to find collaborators with same interest in the US and in Europe. Yeah. But since you are many of you are postgraduate students, maybe I can share some opportunities provided by the number one is WHO IRC. You can go to WHO IRC website. Every year they are providing, especially those who are doing PhD and almost completing the PhD, they are opening fellowship position in the WHO IRC. I think at least two from Indonesia has received this fellowship. I think both of them are currently being become a lecturer in UGM. Dr. Neni, Dr. Dewajani Pramosari, and Dr. Yaya. Yeah, Fatia, I can't remember exactly her full name. At least two from Indonesia has received this fellowship. And it's a good opportunity. Will have the opportunity to do a very high level research with the collaborator there. For the Muslim researcher also, we have IDB, Islamic Developmental Bank, and they're also providing funding for postdoctoral research as well as PhD. My wife was one of the recipient for IDB, and yeah, they are giving quite a good funding for our research. I think these are two opportunities that we can you can explore, right? Oh, okay. So there's a request for me to write down. So one is
So okay, you can, Nur Adisa, you can try. Hopefully, try to apply. Yeah.
yeah. Baik, terima kasih.
Any other question? Ada Prof. Nike, Bu Rita, silakan jika ada pertanyaan. Oke, Bu Rita, Dr. Rita, silakan. Do you have any question? Ya. Mikrofonnya belum.
Oke, terima kasih Prof. Yoni atas kesempatannya untuk ikut serta dalam guest lecture ini. Terima kasih juga pada Prof. Arifin yang telah berbagi ilmu. Pertanyaan saya adalah, telah begitu banyak penelitian miRNA ini untuk sebagai detektor awal atau deteksi dini berbagai penyakit ya dan kanker. Nah, pertanyaannya adalah sampai sejauh mana di Malaysia ya, kebetulan kan Prof. Arifin juga di suatu perguruan tinggi yang kedokteran, apakah hasil-hasil riset yang sudah disampaikan dan cukup menjanjikan ini sudah bisa atau sudah dipergunakan oleh para praktisi di rumah sakit karena kan kita dengar banyak sekali orang Indonesia kalau sakit kanker larinya ke Malaysia gitu. Apakah memang karena di Malaysia sudah diaplikasikan gitu? Maaf, ini pertanyaan awam yang barangkali bisa menambah wawasan untuk saya. Terima kasih.
Terima kasih Bu. So yeah, kebanyakan I think saya jawab di bahasa Melayu ya. Bu ya.
Bahasa Inggris nggak apa-apa, cuman saya kurang lancar berbahasa Inggris.
Kalau di Malaysia ini kebanyakannya kerana miRNA ini masih baru dibandingkan dengan research yang lain, terutamanya kalau clinical itu kebanyakannya menggunakan antibody. Ini sebagai detektor biomarker yang utama. Dan juga kami sudah juga bermula menggunakan indikator genetik iaitu mutation untuk personalized medicine. Jadi kebanyakannya yang sampai ke level clinical application itu lebih kepada situlah di protein apa ni serum biomarker seperti CEA ataupun choreo embryogenic antigen untuk kanser dan juga mutation untuk specific management of cancer patient. MicroRNA masih baru, belum ada yang kerana di Malaysia ni untuk sesuatu biomarker itu menjadi diterima dan di register sebagai rutin test di clinical, dia memerlukan proses yang panjang. Jadi sesuatu candidate microRNA contohnya yang saya dalam projek kami itu baru ditemui 2 biomarker, perlu disambung dengan research di dalam cohort patient yang lebih besar, dan kalau dikonfirm nanti perlu dilakukan satu lagi research oleh NIH, National Institute of Health. Jadi prosesnya agak panjang. Untuk diterima sebagai rutin test itu belum ada yang betul-betul digunakan di clinical ni, tetapi itulah research. Kita buat sekarang, kemungkinan 20-30 tahun lagi barulah akan ada betul-betul aplikasinya di clinical level lah. Dan kita tahu medical people, they work according to protocol, and to develop the protocols, it requires a lot of works lah, and it takes a lot of time. So but research should continue so that we can continue developing our services lah in the future. Right. So tetapi kalau dari segi impactnya kepada praktis dari segi diet, for example, ada beberapa microRNA yang kita tahu berkait rapat dengan certain nutrient. Jadi kita boleh memberikan itu sebagai satu sumber information kepada dietitian, for example. Dietitian boleh memberitahu bahawa sesuatu makanan itu mungkin boleh benefit certain specific condition, specific patient with specific condition, dan dia boleh bercerita, boleh convince the patient through the knowledge based or evidence based health practices. So dengan mengambil suatu nutrien itu boleh menaikkan mekanisme melalui microRNA, dan ini boleh convince the person to change, to have a change of diet lah. So dari segi itu insyaAllah boleh dilakukan dalam waktu yang terdekat. Tetapi contohnya lah, yang satu study saya buat bersama-sama dietitian ialah berkaitan dengan kalau di Malaysia dipanggil daun gajus. I remember saya tak ingat nama saintifiknya, tapi we found that consuming daun gajus ini bagus untuk induce insulin sensitivity and improve the condition lah. So itu boleh dijadikan suatu suatu informasi untuk dietitian promote this kind of food lah, untuk those, especially kepada mereka yang mempunyai masalah diabetes type 2.
Terima kasih informasinya, Profesor.
Terima kasih.
Very interesting. Yes, I think similar thing happen also in Indonesia, that we research, of course, have to be continued. The application will, they have certain procedure to go there, yeah. Okay. Still any other question? Masih ada lagi? Silakan. Jika tak ada lagi, boleh kita akhiri, Dr. Arifin. Tentunya sekali lagi, thank you very much, sebanyak-banyak terima kasih. Alhamdulillah telah membagi very interesting topic for our students today. Jika boleh nih teman-teman student semua on cam ya, kita ambil gambar dulu nih semuanya. Oke, nanti Yasmin yang ambil gambar ya. Please open all of your camera. Nah, Dok Amak, wah, oh ini Mbak Anu ya. Oke, ya.
Baik, untuk teman-teman, izin saya screen capture dulu ya. 1, 2, 3. Baik, kemudian untuk slide kedua, sudah semuanya ternyata. Bisa sekali lagi teman-teman. 1, 2,
Smile ya, smile.
3. Baik, sudah. Terima kasih banyak.
Thank you. Oke, terima kasih sekali lagi
Terima
Dr.
kasih,
Arifin.
Pak.
Terima kasih.
Nanti
Insya Allah.
jika ada permintaan lagi, jangan bosan-bosan nanti.
Insya Allah.
Thank you.
Boleh.
Thank you very
Terima
much
kasih.
from Bogor. Greeting from Bogor to your family also, and
Terima
hopefully
kasih.
we can jumpa lagi.
Ya.
Terima kasih semuanya.
Terima
Wassalamualaikum
kasih.
warahmatullahi wabarakatuh.
Assalamualaikum warahmatullahi wabarakatuh.
Thank you so much.